In this video, we're going to review polymerase chain reaction, otherwise known as PCR. Now, colloquially, people say a PCR reaction, which would be a polymerase chain reaction. So I will use both of those terms, but in this video, we're going to talk about how was PCR invented, how was it originally done, and how can we do this today? So we're going to start with what is PCR, polymerase chain reaction, how does it work, and when is it utilized? So I would make the argument that PCR or polymerase chain reaction is used to amplify specific DNA sequences with guiding primers, which we'll discuss in a minute, that tell you which DNA to amplify. It was developed in 1983 by Carey Mullis, and in 1993, so 10 years later, he and Michael Smith were awarded the Nobel Prize in Chemistry for developing this method. The majority of biochemistry research labs utilize PCR in one form or another. We utilize PCR to amplify genes out of DNA. We use this to make changes to the DNA to study a protein. And so one of the ways that this is used is in order to ask ourselves, what does this do? We need to be able to identify the DNA. So PCR is also the foundation for qPCR, which is one of the different ways that they can measure RNA. So we've seen that in conventional testing. So you might think like a COVID test, the kind that went way up your nose and utilized PCR. This is the same technique as that, although it's a little different, and we'll talk about that when we talk about qPCR later in the semester. So like many things in the 90s, PCR was substantially more difficult. So a PCR cycle has three essential steps. So if we think about PCR, we need to denature our DNA in order to allow the primers to bind. Then we need to create the copy. So when we think about these three steps, these are going to occur at three different temperatures. And so one thing we can think about is that denaturing of DNA has to be at a high temperature, so typically between 95 and 98 degrees Celsius. Primer binding happens anywhere from about 55 all the way up to 68 degrees, and creating the copy happens at anywhere from 65 all the way to 72, give or take. So today we would use a block or an instrument in order to do that. So in the early PCR, there were really two issues. In their tube, where you have your liquid, if you heat it up to 98 degrees, all of your water is going to evaporate. Once you start having evaporation issues, you can actually massively damage your enzyme. So we have this tube, and this contains our sample. And so this sample is going to evaporate, frankly, to the top of the tube. So one way that they used to fix that is they would put a little layer of oil on top. Conventionally, people don't do this anymore. It was kind of a lot back then. So that's one. But the other thing is, if you look at this graphic over here, so this is, I don't want to call it an original PCR block, but one way that we did this, we have a 98 degree water bath. We have in this case, we'll just decide that it's 58 degrees and a 68 degrees. And so the scientist would come and set their tube in here for the given amount of time. Then they would take it out and put it here and do something. And then they would move it here. So there were two issues with this. One, super time consuming because you have to monitor every single step. The other issue is that when you heat an enzyme to 98 degrees Celsius, that enzyme is going to denature. And so in upcoming slides, we will look at how that works. And so we'll look at how they change the thermal stability of these enzymes. So a PCR overview. We are going to amplify specific DNA using targeted primers. So the way I like to think about this is PCR is a Xerox machine, but for DNA. So we know, as previously discussed, how DNA is replicated with polymerase 3 and a complex, and you can basically have that RNA primer and it will specifically copy until it gets to the end. This is the same thing. So if you have a book and you want to copy page 105, you would copy page 104 to 106, and then once you make that copy of page 105, you could just put that through the copier and make a bunch of copies of that. So the way this works is what we're going to have here. And I'm going to move this up a little bit. So here's our original DNA shown on the left, and we have nucleotides and primers. Now these two primers are the same color, so we are going to annotate one as the forward and one as the reverse. So in order for you to have two primers, because one can bind on the 5' end here and one can bind here. So this one is typically called the forward because it copies the sense strand, and this one is considered the reverse because it's going to copy the antisense strand. So this step here would be your denature. This step here is the annealing. Here is the extension. And then with this graphic on the very end, this is your repetition. So if you have 5 pieces of DNA at the end of one cycle, each one of those should have been made into 2. So if you start with 5 copies, after cycle 1, you should have 10 copies. After cycle 2, you should have 20 copies. You can see that this is going to be an exponential amount. So if you were to think about how much DNA is made, down here we have cycles, and on this y-axis we're going to have a random absorption. And so what you should see is something that eventually looks like this. And there are some pretty standard amounts where we can see it is the concentration of DNA multiplied by 2 raised to the number of cycles. CYC minus 1. And so this will give you the concentration at the end of that. And so when we think about PCR, what we're really doing is taking the polymerase 1 or 3, either one of these, and so different enzymes use either 1 or 3 depending on where they come from. And then in a tube, you're going to have nucleotides, all of the enzyme components in your template, and you're going to run this cycle. I tend to run mine for 30 cycles. And so what we see here is that this repetition is going to make specific copies. So there are a couple of things we kind of want to think about. At the end of the first cycle, there's nothing to stop this extension here. And so that extension goes on until time stops. And so you can see that over time, you'll continue to get some of these extra edges, but once the primer copies continue to move, so in this sample here that I made in a box, when the reverse primer binds and it goes across, it turns out that it can only go all the way to the other primer because there's nothing left for it to copy. So over time, you will get a predominantly the right amount or the right length as your 2 primers. So we've talked about these 3 steps, and so we can see that denaturing separates DNA. Annealing is going to allow the primer to bind. An extension is going to copy the gene of interest. And so once you copy this gene of interest, you can repeat this cycle. In this case, we're going to say 30x, and that's going to give you a high concentration that is based on your starting material. So in this reaction tube, what all do we have to have in there? So if we have a reaction tube, we need to have a couple of things. You need to have your gene of interest. And this can come from a different plasmid or from genomic DNA. This is your starting material. So we need that. We are going to need some version of a polymerase. And we will talk about how you could pick one of those in a second. Then you're also going to need nucleotides. And these are things like TTP, the ATP, the GTP, the CTP. So these are deoxy triphosphate or triphosphate of the different bases. Generally, people just call them the NTPs or deoxynucleotide triphosphates. So you need those, and they need to be stable. And then there tends to be some type of a buffer. This often already, it could contain your nucleotides depending on your mix, but this is going to have some salts. It's going to maintain the pH and would provide any cofactors that your polymerase requires. And so also you're going to need to probably add some water. And the water here is going to give you a final concentration. So this reaction goes anywhere from 25 microliters all the way up to, I would say, 100 microliters, kind of depending on what your final goal is for your reaction. But these are the reagents. And so this buffer is going to have the required magnesium 2+, in addition to a few other things. We forgot to add primers. So you also need a primer, and this would contain both your forward and reverse. So next, let's really think about what each of these different things are. So your primer. A primer is a short, meaning 12 to 25, although I've used things all the way up to 40 nucleotides, base that binds to the denatured DNA. So in this case, we have the forward on the bottom because it is going to copy in the sense direction, as well as the reverse, which is in the antisense. So there's a couple of things that people want to think about. So things you want to think about. So in this case, these primers don't have a lot of GC. So some people find that they want primers with high GC content, although not too much because then they don't denature. But a lot of people want to have a GC clamp at the end because then you are assured that it's going to bind in the right place. So the longer it is, the more specific the binding. The downside is the longer it is, the harder it is to get it to denature. So your primers, you can purchase these from a company. And so you buy these and they are specific to the gene. So in all cases for PCR, in terms of amplification, you need to have two primers. One on the forward strand and one on the reverse strand. So when we think about the enzyme or the polymerase, so your polymerase, there's a couple of things you want to think about. Fidelity, otherwise known as proofreading. So we know that we need our DNA to be accurate. So there's always a play between speed and proofreading. We would like it to amplify quite quickly, but we would also like it to be super accurate. And so the yield, we also want to make sure that it is able to continue to run through all the cycles. So it's not going to stop working. So down here, these are different brand names. So TAC is a polymerase that is kind of thought of as the cheapest. It is very affordable, doesn't have the greatest fidelity, but for a lot of things it works really well. Fusion and PFU Ultra tend to have super high fidelity and quite quick speeds. So when you order your enzymes, so enzymes are purchased from a company, and in that documentation, when they mail it to you, it tells you how fast, the best cycles. You can think about it kind of like when you order something and it says things like, in order to cook this meal, you need to put it in the microwave for 3 seconds, stir, put it back in. You can make other changes and you may over time decide that you prefer your meals to be quite different, but overall, we're going to follow the directions. So that is one way that this can be set up. So what tips and tricks are there for this reaction? So if we draw out a reaction, so in my lab notebook, in my research lab, here is a given cycle. So we have 98 degrees for 30 seconds. Then we have 98 degrees for 10 seconds, 62 degrees for 20 seconds, and then 72 for 2 minutes, followed by a 72 for 5 minutes. And these three are going to be repeated 30 times. So there's a couple of things that we want to think about. This first step here is just an initial denaturation. Basically saying we want all the DNA to denature the first time. Some enzymes require a hot start, meaning you're going to kind of cook them to get them up to speed. So in this repeating step, this 10 seconds is a denature. The 20 seconds is the annealing. This is our extension. And this down here is a final extension. This allows any pieces that are maybe not fully created to be completed. Then we're going to hold at 4 degrees for forever. This is essentially a way to keep your DNA stable and not repeating through this cycle. So if we look at this, you might think, hey, I'm not sure we need this right after the initial denaturation. That is true, but you do need it in terms of every other cycle. So typically the initial denaturation kind of sets that up. The other thing when we think about a PCR block, so a PCR block, I'm going to kind of sketch one out. And the one in my lab kind of looks like a giant box. Here at the top, if you were to open it, has a series of places where you can set your small tubes. And on the front, it has a touch screen where you can design this program. So you can set this up and you can change it. So the extension, both the temperature and the time are dependent. So the temperature is based on the enzyme. The time is based on the gene size. So if you have a short piece of DNA, it would be a shorter extension time because at some point you need it to copy all of the bases in your gene, but it doesn't need to just keep copying just because. Additionally, for every primer that binds, you want to have a polymerase attached to it. So there are concentration dependencies. So some tips and tricks. One, you want to make sure that you follow the specs. So you want to follow the specs because if you're not following the specs, then it's hard to know what your enzyme needs in terms of both reagents and everything else. Two, you want to keep your reagents cold for stability. So we want to keep them cold, i.e. on ice, because your DNTPs will degrade. So if you think about the hydrolysis of that phosphate bond, that can lead to problems over time. So you want to keep them cold, you want to keep them stable. And three, you want to actually make sure that you are analyzing the results correctly. So we can see that here. So there's two things. How do we actually analyze this? We're going to take an agarose gel. So this is agarose. And so in this lane, we're going to have a ladder. And we have a ladder so that we know what it should look like. So we'll have 1, 2, 3, 4, 5, 6. So this is our ladder, and we're going to put our PCR product in these two lanes. And so in lane A and B, these are going to be two different reactions. So our goal is to amplify. We're going to actually have a third. Let's see. A 2800 base pair gene. So here are three different types of results that you could get. So in lane A, it's going to work the best. We are going to get a really clear band, just one band, that's all. So in lane B, we're going to get some of this, but we're going to get some other things. And our largest band is going to be at 1 kb. And in lane C, it's not going to work at all. And what we're going to see down here at a very low molecular weight, these are primers. So if your reaction works, all of your primers should be used. If nothing works, all you see left are primers. So in lane A, it works well. And you can use that PCR product. In lane B, it did not work well. So you can, and I'll draw it in red, you could, using a different apparatus, you could actually just excise or cut out that band and move forward. In lane C, it did not work at all. So in lane C, what do? One, you're probably going to want to try to run it again, mostly because maybe you forgot to add something. Or you're working with really small volumes of things like 1 microliter, maybe it just didn't get in the tube. Alternatively, it could have also your enzyme, something else could have degraded. So we're going to have to look back at that over time. So in this case, we want to always run a gel in order to do that. And so you would visualize that using something like ethidium bromide or a safe stain. So this has predominantly talked about amplification of a gene in order to use that in molecular cloning. But what else could you use that? So as we'll see later, we can sequence DNA or RNA. This uses one primer. And we will look at this technique in detail. When we think about forensics using LRRs or SRR, which are long repeat regions or short repeat regions, all of these are PCR based. So you can use a specific primer in order to amplify that for known conditions. So one way that they test for infectious disease is they use a group of primers and then look for amplification of specific genes. So qPCR, RT-PCR, which is this measures RNA concentration, RNA. But what we can see is this is different ways that we can look for that. So PCR is a really common technique in molecular biology, biochemistry, biophysics, because it's going to allow us to study and manipulate DNA. And we will come back to PCR repeatedly in many of the different techniques that we look at.